Okay, Claudia was supposed to introduce me, but she couldn't be here at the last minute, so I'm going to introduce myself. So I'm Sue Trumbore. I'm one of the original founders of the Earth System Science Department here, but I'm only 20% appointed now, so I'm here once every 3 years to teach a class. And so this is why I'm here now. So it's always great to come back. And usually I come back more frequently, but during the pandemic, of course, that wasn't too easy. So I'm going to talk today about some of the stuff that we've been doing, and it happens to be a collaboration with UCI through the Keck AMS lab, with especially Xiaomei Xu and Jim Anderson and Steve Allison and Liz Ray. So as part of this in the recent past. So I'll explain a little bit about the project and what we were trying to do, and it's not my favorite isotope, so it's gotta be cool. Okay, so the reason we're trying to, well, the research we're trying to do is to determine how fast carbon moves through land ecosystems. And that's important because it's the source of the largest uncertainties in the land carbon balance. So this is one of the CF6 slides that shows just how bad our understanding of the land carbon cycle is. It's looking at the fully coupled models and the spread on the models. So the blue is the fully coupled predictions. So after 140 years, the average model is predicting 635 petagrams of carbon will go into the lower biosphere, which would be doubling the size of the pre-industrial vegetation pool. So it's a huge change. The green is what's driven by CO2 fertilization in those models, and the red, which makes the blue a little bit lower than the green, is the carbon loss that's driven by warming and decomposition feedbacks. But really, all of these things are uncertain because we don't understand, if we want to understand carbon uptake by CO2 fertilization, we have to know how long it is before that CO2, the extra CO2 that is fixed, how long does it stay in ecosystems before it gets respired back to the atmosphere. And that really determines the size of the land carbon sink over CO2 fertilization or other kinds of fertilization. And when we think about carbon loss driven by warming, we want to know what portion of the overall pool of carbon in terrestrial ecosystems might be vulnerable on the timescales of a century. And so the models are doing their best. They got their best understanding, and that's why the best understanding isn't very good about now. So that's why these models probably overestimate carbon sinks and maybe also carbon sources. So the measure that we have to test these models, and this is one of the problems, so the measure that we most often use to test models of carbon is, are we getting the stocks right, and are we getting the fluxes right? And that's basically, if you get the stocks and the fluxes, then you should get the turnover time right, which is, if you think of it as a homogeneous system, it's just what geochemists love to do, take a box, measure the inputs and the outputs, and then calculate the turnover time, which would be the response time of that system going forward. But we know that it's not that simple when we look at terrestrial ecosystems. There's sort of one way in for carbon, which is photosynthesis by the plants. Then how fast or how long carbon resides in that system before going back to the atmosphere really depends on a whole bunch of processes that we don't understand very well in ecosystems. So, it could be allocated to either inside the plant to respiration, that lets the plant maintain its metabolism. It could be allocated to growth of tissues like leaves or fine roots that might live for a couple of years or less than a year. Or it could be allocated to stems that might stick around for 100 years. And once that material is dies, it goes into the soil and becomes the source of microbial plus faunal decomposition processes that ultimately bring that CO2 back to the atmosphere. But the time scale depends on what kind of material it is, what kind of microbes you have, whether you have minerals that might stabilize some of that material for a certain amount of time. And so you can see that it's a complicated system. It's not a homogeneous system, it's very heterogeneous, and every one of those processes from allocation within the plant to microbial respiration have climatic interactions with climate and biology, which makes it hard. So what we're trying to do in C14 constraint is to really increase the number of metrics that we have to compare with the models, and using a focus of the timescales and what we can measure with radiocarbon. So if you think of a non-homogeneous system, so we have a heterogeneous system with cascading carbon flow through pools that may cycle fast or slow or very slow, then the system age is the age of the carbon in the system. So it's the time that the carbon in any one of those boxes, the time elapsed from the time it was fixed by the plant to what it is now in that pool. And then to measure, to get the bulk, we're just weighting that by how much mass is in each pool. And typically, as the geochemists know, the biggest pools are the ones that cycle the slowest. That's why they're the biggest pools. And so anything that we measure in bulk soil to measure the age of the carbon that's in there is going to be weighted towards the passive pools, and I emphasize the more stored pools. So the other metric that we're looking at is what we call the transit time, which is looking at the age of the carbon that's leaving the system. So it's how much time has elapsed since the carbon was fixed to the time it's respired back to the atmosphere. And we feel like, especially on timescales of the next 100 years, this is the metric that means the most for comparison with models. If the models are getting that right, it doesn't matter if they get these old pools that aren't going to do anything right, because it's just the wrong timescale. So for the system age, what we measure is the radiocarbon in soils, bulk, and we try to separate it maybe into fast, slow, and passive cycling fractions and measure the radiocarbon in those. For the transit time, we're measuring the age of carbon that's being respired from the soil. And so there we're letting the microbes tell us what they're eating, which is, the best measure of what we want to know, which is the age of the carbon that's going back into the atmosphere. And they shouldn't be the same, right? So because the transit time is the respired carbon, it's weighted towards the faster cycling pools that contribute the most to the flux, whereas the age will be weighted towards the massive pools that are the slowest ones. So we don't expect those two numbers to be equal because it's not a homogeneous system. The transit time should be shorter than the system age. And that's just, this is kind of what we would expect. It's not, if you have even a simple box model, you're going to get a distribution of ages. And so if we look at the age of the carbon being respired, the amount of carbon being respired versus the time since it was fixed, this would be for the transit time. We would have contributions from the plants of the rhizosphere respiration, the sort of plant metabolism, which especially in big organisms like trees is not necessarily when you just fixed gets respired. And then you have a heterotrophic respiration, the microbial respiration that includes the age of the material. Like if you have a 100-year-old tree stem that dies and decomposes, that's going to show up as a long tail in the respiration distribution. So how do we tell time in the carbon cycle? We use radiocarbon, and there's two kinds of radiocarbon. There's the cosmogenic radiocarbon that's produced continuously in the atmosphere, and that we use for dating things that are older. Once it's made in the atmosphere, it's entering the carbon cycle from the atmosphere, and ultimately will decay again radioactively with a half-life of about 5,700 years back to nitrogen-14. So we have carbon-14 only because it's continuously being created and then it decays away, and it partitions itself between the atmosphere, ocean, land, and we can use the fact that plants take it up, make pieces of animals that leave bones or whatever that we can date. And as long as it's a closed system, we can use how much radioactive decay occurred and infer that age. But I'm going to talk mostly about bomb radiocarbon, which is the other big source of radiocarbon, which was created in the most of it was created in the early 1960s with atmospheric testing of nuclear weapons, like this nice test here. And what you can see, this is a graph of the radiocarbon and atmospheric CO2 over time, which has been records that were kept by lots and lots of really dedicated scientists over the years who kept these records going so that we would know what the atmospheric radiocarbon was through time. And what you can see is in the early 1960s, you see a sudden rise. The units here are delta C14, which is a lot of the stable isotope delta C13. It's the ratio of our sample to a standard, which the standard is now fixed in time, as well as isotopic signatures. So it's the pre-industrial wood, basically. And then you subtract 1 and multiply times 1,000, so it's in units of per mil. So 0 on the scale would be equivalent to the pre-industrial atmospheric CO2 value, and then 1,000 would be a doubling of the atmospheric CO2. So in the northern hemisphere, the radiocarbon in the atmosphere was almost doubled. And since we can measure it very sensitively, this is an enormous global isotope tracer for the whole carbon cycle that a number of people have spent their careers chasing, like me. And as you'll see, chasing it during your career is actually advantageous because you can make time series that really help you with the modeling. So, okay, and then I'm going to cheat a little bit and I'll show you mostly this delta C14 unit. So if you just blow up the last decade or so of the radiocarbon curve, what you can see is that the decline is almost linear. So it's declining in time because of mixing of the bomb C14 into the oceans and into the uptake by the biosphere. But over time, that rate of decline slowed, and that's partly because things are starting to come back out of the biosphere and recycle. But it continues to decline because we're putting so much fossil fuel and CO2 in the atmosphere. So we're diluting everything in the atmosphere with carbon-12 that has no carbon-14, and that's what's actually driving the current decline. So we've actually just passed the level of the pre-industrial atmosphere, and we're going down, but as long as we keep pumping fossil fuel, we've still got a good tracer. So, but anyway, the delta C14 is the atmospheric curve tells us what the radiocarbon signature of carbon fixed in any given year would be. And so if I go out and I measure CO2, so the blue dot above the curve there, would be a typical measurement that I might make of a CO2 coming out of the soil or a soil incubation, and it's above that. So that means that the material, the carbon there was fixed, had to have been fixed several years previously in order to have a higher radiocarbon than the year that I'm looking at. And so I'm just expressing that as a delta C14, so it's the amount, it's the difference between my measurement and the year, the atmospheric C14 O2 in the year I took the sample. And I'm doing that because we have samples that were taken over a decade, and the reason we get away with the delta C14 is because it's been relatively constant, the rate of decline in the atmosphere over that time. Okay, so here's something that you can use in your next cocktail party. When we think about the allocation within trees, one of the things is, how long, we often, at least I often thought that, that's all very fast processes, but one of the projects we had was with a group of maple syrup producers in Canada who they wanted to understand how old the maple syrup is. It's just one of those questions you ask yourself. Or when you measure carbon-14, this is a question you ask yourself all the time. It's like, how old is that carbon? So we measured a bunch of maple syrup. These are all different stands. So the syrup itself has about 2% carbon, so you can see the thing dripping out of the tap. It drips out of the tap because you can only do maple syrup harvesting when it's freezing at night and warm during the day. So that actually is what's kind of pumping the stuff out of the tree if you have a spigot in it. Anyway, if you evaporate all that and measure what's in it, the radiocarbon content was about 35 per mil more, or 15 per mil more than the atmosphere. The atmosphere is declining at a rate of about 4 per mil per year, so that gives us something between 3 and 5 years. As the time elapsed since the carbon that was fixed from the atmosphere and ended up going up the tree and then sap that would become leaves. And the other thing, so this is kind of a simple age model where we're just saying what's the mean time elapsed between when that carbon was fixed and when it ended up in your maple syrup. So that tells you something about the vulnerability of the maple syrup crop, maybe, to a bad year. If it's coming from storage reserves that are there for years, maybe you don't have to worry about a particularly bad year for next year's maple syrup crop. The other thing we did was this. There's this famous kind of McNeely curve where Roger McNeely had collected maple leaves and he had his own bomb curve through the bomb period. And that was always slightly above the atmospheric curve, and it was kind of puzzling as to why, was it a nuclear facility nearby or something like that. But if you do a mass balance and you say, well, some of that leaf had to grow, the leaf bud and maybe the petiole and parts of the leaf itself had to grow from the stored carbon before it could become a net source of its own carbon, then the age of the carbon in the leaf should be, then if you just do a mass balance, you could say part of this is reserves, and something like 70–80% is from recent photosynthesis after the leaf was formed, but it's made out of this old material too. And I have no idea why in the 1980s things weren't there, but that's probably somebody's test or something. But we think we can explain this. Okay, so far, radiocarbon tells us the time elapsed since carbon was fixed from the atmosphere. A number of people make the mistake, including me as a younger scientist, to say that that's equivalent to like a decomposition rate, but it's not. It's including all the time spent in the plant. And you've seen that time spent in the plant, especially in trees, can be significant. So, and the other thing is that we need models to interpret the radiocarbon data from these heterogeneous systems. So maple syrup, we used a one-pool model, but probably it's not that easy. And the metrics that we use and compare are going to depend on the model that we're using to interpret our data. So, but the upside of that is that, all existing carbon cycle models will predict radiocarbon, and we can compare those with our measurements. So we can measure and model the same thing, which is something that doesn't often happen in the model world. So the goals of the C14 constraint project were to, increase the use of radiocarbon because we feel like it's this tremendous tool that nobody's using partly because it's hard to interpret, even for people who are experts. And so we wanted to do 3 things really. First is to sort of gather all the radiocarbon data that existed and make some data products that we could upscale. We wanted to provide tools and approaches to sort of facilitate this model data intercomparison in ways that people could easily use them. And then we also wanted to generate new understanding. So I'll go through just a little bit of that, and we're kind of stuck at one point, so I want to get advice also from people. So the first thing that we did was this bring together all the existing radiocarbon data that we could find. And this wasn't just our group at, not even the C14 constraint group. We joined forces with the PAL Center and a lot of different researchers to create this sort of open source, community-based database. There's a bunch of people that you may not know. One's now at the people in Washington, as we're anyway, we all would have these hackathons and kind of get the idea of the database worked out, and we compiled it, and you can go to the website, soilradiocarbon.org, but anyway, it's not just bulk radiocarbon data. It is a function of depth from soils. It includes radiocarbon from surface fluxes, fractions, CO2 in air pore space, anything that people have been measuring, DOC, everything's in there. So if you want to find data, it might be there. Our only request is that if you use a lot of data, you can either contribute some data or help us fill out some records so that we can keep it going without much funding. This is what's currently in the database. It's grown a lot. So when we produced our first version, it had probably about half this number or even less in profiles. We added a lot of data from the boreal and Arctic, and through collaborators we've been adding as we go. So the first data product, I'm not going to talk about much. So this was Zheng Shi, who was a postdoc here with Jim and Steve Allison. And the idea was to use the bulk radiocarbon data and profiles, use random forests to kind of relate those values to climatic and soil properties, and then use that to upscale and make the first sort of global data product, radiocarbon map of carbon in soils. And that paper showed that the models pretty much, that the carbon-14 values in soils were lower than the ones that the models were predicting. So this is why we think that the models are all storing too much carbon in soils, because they have been responding on these sort of decadal timescales with too much carbon. So that's looking at the system age. So the product I want to talk to you about now is the second data product, which is this, which is what we're working on now and where we're a little stuck, where we look at the global transit time. So that is the global system age distribution. So the transit time is very low tech up until when you get to the accelerator. So you can see, hopefully inside the jars, there's soil in jars, and then we're measuring the soil in the jar, that's the bulk radiocarbon, but then we're also measuring the CO2 that the microbes in the soil are respiring, and we collect that, and we measure that. So, and that's kind of what we're saying approximately we could use to estimate the transit time, this fluctuated C14 or CO2. And there we have less data in the database. So the one I showed you before was any place that has a C14 number had a dot. These are just the places where there's respired CO2, which is actually way more than I thought. But it's not a huge amount if you think we want to upscale globally when there are large data gaps. So again, I'm going to present these delta-delta C14 numbers, which is the difference between either the bulk soil or the microbial or respired CO2 and the atmosphere in the year that the measurements were made. So I'm going to spend a little time on this one. So this is the delta-delta C14. So it's the difference from the atmosphere. So 0 means it's the same as the atmosphere the year it was collected. And on the x-axis is the bulk soil organic carbon. So you see values going from 0, so positive values, I kind of made little lines at the zeros. Positive values means there's bomb carbon-14. That means that the majority of that carbon was fixed from the atmosphere in the last 60 years. The negative values mean that there's been enough time for radioactive decay since that carbon was fixed from the atmosphere. Minus 500 per mil would be one half-life, so that would be about 5,700 years, more the time scale. And what you can see is the points largely scatter above the 1-to-1 line, which means that which is the y-axis is the microbial respired CO2 has higher radiocarbon values than the bulk soil organic carbon from which it was being respired. So that's what we expect. We expect that the fluctuated carbon being lost, we're eating the youngest and yummiest stuff, so we're respiring that. It's younger than the majority of the carbon that's in the soil. And that's what you see by and large there. There's a little cluster of points in the bomb period that's lying to on the downside of the 1-to-1 line. And those can be explained too. It just means that there's very fast carbon covering all those. But what I want you to focus on here is the color, so the color coded by what the depth is, and what you can see is that, most of the bomb carbon, you're sitting close to the 1-to-1 line, and if you get to the deeper and deeper you get in the soil, sort of the further off the 1-to-1 line you are, where you're respiring much younger carbon than is there. So very little of the carbon that makes it out of the soil is respired in the deep soil. It's contributing maybe a few % of what's coming out of the surface, so we're going to ignore it. And I'm just going to concentrate on the next slides of this, and I'm sorry I changed color scales on you. So if we look at surface litter, these are just all the data we have. They're mostly clustered around the 1-to-1 line. They're mostly above the 0 line here. The color now means mean annual temperature, so the warmer the color, the warmer the temperature of the site where the data are from. And what you can see is surface litter is sort of scattered around 1-to-1. The mineral soils are on a different scale. They have a larger range, but you can see that the higher temperature, so the orange color, is mostly on the 1-to-1 line, but as you get colder and colder, you fall further and further off of it. So these are just all the data. So what I did here was average the data from one site, so that we're not overemphasizing one place. If there are more than one point, that means we might have multiple years of data from one site. And that might explain why they're not always sitting on top of each other. So on the left is the surface litter, and on the right is the 0 to 20 centimeter mineral soil. What you can see, or what I'm going to make you see, is that there's kind of, what happens is at the high temperatures, so temperatures tropical kind of latitudes, the difference between what's being respired and the 0 is close to 0. You do see the different colors there are it's the mean annual precipitation divided by PET, so potential evapotranspiration. So it's a measure of how wet it is. And you can see there's also probably, in addition to temperature, some moisture influence on what we're seeing. But you don't have a lot of data, especially in the surface layer. And then as you go towards lower and lower temperatures, higher and higher latitudes, you're getting values that are higher delta C14. And then there's some point where they turn around and they go back lower again. And it's basically going through the bomb curve where you're getting older and older carbon, but then if you start to respire some pre-bomb carbon along with the older carbon, you ultimately go below 0. And so that's what we think is happening, but how do we turn that into quantitative information? So we do the simplest thing, which is this one pool model. And the big problem with the one pool model is that when you, this is here's the year versus the delta C14, the atmosphere is the black curve on the left, and the blue curve and the red curve are what a one pool model would give you for the radiocarbon change over time in a pool with either 13 years turnover time, so that's the faster one, the red curve, or 36 years turnover time, which is the blue curve. And you can see that if you make an observation in the year 2000, you get exactly the same number. And so this is our problem. We have the delta C14 for a certain year, but we don't know which of those models. We have equifinality for our models. So we have more than one model that can give us the answer. And we have to have additional information to say which one is the correct one. And so on the right, I've just got what the radiocarbon delta C14 is for different years with different transit times. And you can see it's a problem that doesn't go away. If you have multiple time points, you might be able to start to tell the difference. But since, things were, anyway, what we did was we looked here and we said, well, there's some break point where, turnover times or transit times are increasing with decreasing temperature, and then they have to keep increasing. So at some point we're switching from the fast part of this curve to the slow part of this curve, from one to the other. And so for this, we just made a guess and said we're going to set that at 0 degrees. And if you do that, these are the transit times that you get for the different radiocarbon values. And again, versus mean annual temperature. And, it gives you something that makes a lot of sense. They're plotted on a log scale. So they look a little better than they would if I plotted them on a linear scale. I'm not arguing that this is the right thing to do, but it was what we thought of first and why we're stuck. Because, we say, we'll just use random forms, we'll scale this up, we've got a nice relationship with temperature, precipitation's in there too, it'll be great. But now I'm kind of stuck. But anyway, years to decades in the tropics for transit times, centuries when you get to high latitudes, I think that actually agrees fairly well with the other way of estimating this, which is the turnover time. So there's a couple of papers from another group in our institute that's been doing these data upscaling things where they take the carbon inventory and divide by GPP. And if you take the carbon inventory and divide by GPP, or gross primary production, that's the total ecosystem stock of trees, soils, everything. And they divide by gross primary productivity. So the respiration is in there along with everything else. And this is the number that they get. And it actually agrees pretty well with the numbers we're getting. But I think it has to be an accident. Or if it's not an accident, it's the long-term mean of everything, and maybe it's not so bad. But anyway. Well, we're going to explore that further because we're in the same institute. So just to summarize the main points, so far the climate is a major control of the transit time, so we do see that it is climate, things get older as temperature gets cooler, and that's not a surprise. The results are in broad agreement with these turnover times, and the transit time really should be compared with the turnover time in terms of they should be close to each other. But we also know that the one pool model is probably not a good approximation because, from the 1-to-1 line, as you went further away from the equator, you got further off the 1-to-1 line. So the next thing we did, and this is why it's really good to be an old scientist, is I've been going to Harvard Forest since I first started at UCI, so since the 1990s. And so I have measurements of the respired CO2, which are the pink dots, and the bulk radiocarbon, which are the blue dots. So the first measurements were from archived soils that we got from people at Harvard Forest, and then Julia Gadinsky's thesis was like the very first set of points there, and then the second set was a postdoc named Robert Borkin, who worked with our data today. And then the last one was right before I came here. I went to Harvard went to Harvard and gave 2 talks. One of the talks was at Harvard Forest, and so I made them take me out, and I got samples. And Xiaomei ran them. Well, we did the incubations when I first got here. Xiaomei ran them. We don't have the data for the bulk yet, but I'm going to make predictions here. So, because maybe in the next few weeks we'll get those answers. So, what we need then is we need a model. Remember, we can't interpret these results without a model. We're using a very simple model where you have 2 pools, a fast and a slow pool. You have inputs. There's some parameter that says how much of the inputs goes to each pool, and then you have a decomposition rate for each pool. And this is all implemented in a package called SoilR, where if you want to make any kind of box score models, it's a really nice tool. It's just a matrix-based manipulation of the things. And anyway, if you want to play with one, I have a Shiny app that lets you play with different scenarios. So this is what you kind of see, especially if you use the Shiny app. The pink, so the 2 pool turnover times are, and the trajectories for those 2 pools are the light blue homogeneous pool and the light green homogeneous pool, those trajectories. And then you basically have this gamma value that's telling you how much is going to each place, and you have the flux weighting. So the respired CO2 curve there is just the flux-weighted combination of those 2 curves, and the bulk soil is sitting close to the older pool because that's the mass-weighted combination of those 2 curves. And so they fit the data pretty well. I used again, you can do a Markov chain, Monte Carlo, and get errors. This is all stuff I've learned how to do from my students. And what we see is I've got the surface layer on the left and the horizon on the right, and actually that last point really makes a difference in terms of what we see. And what you see is big differences between the 2, especially for the bulk carbon, and the bulk carbon is close to the 1-to-1 line, so it's almost a one pool could conceivably give you the same answer as the 2-pool model. And it's really important to get that 2023 data point for the A horizon. But what you can see is that if we just looked at the blue points, and we had a single year, and we had taken the surface litter and the A horizon and just done that 1-pool model, we would have said they had the same transit time. But looking at the time series, we see that they don't. And so one of the really nice things that Carlos's SoilR package has is he lets you calculate from your model what the transit time distribution is. So what's the distribution of ages of respired carbon that are coming from the model that best fits the data. So here, like the mean transit time for the surface layer is 5.9 years. The mean transit time for the A horizon CO2, even though it has very similar value, is 106 years, based on fitting both the bulk soil and the respired CO2 data. The medians are similarly quite different. They're not, they're long-tailed distributions. There's small amounts of carbon coming from very old materials that's contributing to the respired CO2. And if we did the 1-pool model, we could get a pretty good, so we had either 5.8 or 85 years is our two possible solutions for the one pool model. For the surface litter, when I did it that way. But then with the A horizon, I would have said it's either 7.5 or 7 years, but with this long time series where we have much better constraints on the data, it's sort of somewhere in between, and that's where I'm stuck right now, because we don't quite know what to do with the A horizons at this point, because the one pool model is not the best way to estimate what's going on. But I guess if I had to use one, I would say the 70-year was the better one to use, because it's closer to the mean transit time that's picked by the two-pool model. Okay, so then we have to ask, like, why is the transit time so much longer for the A horizon? This is just the top soil. Why is it so much longer than the litter that's decomposing on the surface? So it could be, I mean, we have measurements of the mass loss for both root litter and surface litter, and they're not that different from each other. So this is just, calculating the mass loss, and this is kind of equivalent to what we're seeing in Harvard Forest for the surface litter. But why is it so much slower in the mineral soil when we do these kinds of experiments that the roots aren't decomposing that much slower? So it's not that, it's not climate, because it's the same in the top 10 centimeters as it is at the surface. To first order. So, it could be that we have some pre-aging of materials. So, what's dying might be older. And in fact, that's true. So Julia Gadinsky, as part of her PhD thesis all those years ago, found that if we just picked live roots out of the soil and measured the radiocarbon content, the reason she did it was she couldn't explain the radiocarbon values she was seeing in this soil organic matter at the time. And she had to infer some kind of time lag for the carbon entering the soil. And this is just another result that shows the same thing really nicely, because they had archived soils that were collected at different times during the bottom period, and the 8-year turnover time, one pool model fits it perfectly. So that's saying that the carbon in fine roots that most, anybody who looks at roots with many rhizotrons would say they're turning over within months, maybe a couple of years, but certainly not that long. So the root people really didn't like this result. And one of my students, many years later, Emily Solly, went to Switzerland, and they said, well, why don't you just count the rings in the roots, and then you know how old the root is, and then you can measure the radiocarbon. And so she did that, and this is what she found. The root age based on growth rings was much younger than the root age based on radiocarbon, especially, when you get to colder regions and things like this. So basically, what we think now is that new roots are mostly grown from older carbon stores in the tree, but not always. And that's really kind of where anyway, it's an ongoing research question. We're trying to use the age of roots, new grown roots, to say something about how the trees are allocating their carbon. Because it's the furthest away from the leaves, and so it's the place where you need the biggest buffer. Because if there's a big amount of above ground demand, new carbon is not always getting to the root. So, okay. And so if that's true, then we should be able to look into our data and say, well, this is true for woody roots, but for grass roots, it's not true. The grass roots are grown. When we measure their C14, they're what they should be. They're close to the atmosphere. And so this is a little hard to see, but one of my students did, these are like 20 different forests and 20 different grasslands that were incubated. The atmospheric curve is the black on the right there. Sampled at 2 different times, and what you can see is that the brackets are showing you the difference, the delta C14 for the grassland, which is blue, versus the forest on the right, and I hope you can see the bracket on is, the forests are higher than the grassland. So maybe this pre-aging of woody flying roots is, so we have old carbon entering and then it takes not so long to decompose. And that's basically what's giving us this C14 signal that we have. So it's not decomposition rate, it's the whole ecosystem flow of carbon that we're measuring. So part of it is probably this, that the grasslands and the forests are different, but they're not different enough to explain a 20-year difference in the transit time. So this is the topic of another one of my students' PhDs, who's finishing right now, but these are transit time distribution curves. I apologize because they're plotted on a log scale, so they look weird. But if you just look at the vertical bars, the solid lines are the mean transit times for 3 different soils that have the same climate, same vegetation, but all it's different is their mineralogy because they're on different parent materials. What you can see is there's quite a difference that's related to the mineralogy of the soil. So that's another reason that mineral stabilization processes could partly be responsible, since the slow pool is contributing to what's being respired, to what we're measuring that's being respired. It might be 10 or 20% of the flux that we're measuring, but it's still there, and if it has a very different age, then it influences our estimates of transit times. So we don't have an answer yet. And maybe in the interest of time, I'll get through this, but we've been looking at other fractions of organic matter that other people have used. One of the problems is we can't isolate something in the soil itself that is the fast pool. People have been trying to do it, but the one that everybody's fixated on is this low density material that's plant material. It's not associated with minerals. It's the youngest stuff there. But it actually looks a lot like the respired CO2 that we're measuring. And so it also has some fast and slow carbon in it, just as the minerals have fast and slow carbon in them. We just can't isolate those. We can model them, but we can't isolate them. So the work remaining here is, what are we, our upscaling is kind of falling apart because we need to do these 2 pool models, but we can only really do them when we have a time series and be confident of what we're doing. The 1 pool model is not really a good approximation, especially at colder temperatures and greater depths. It's good for the tropics. And temperature is clearly not the only factor. We see soil mineralogy, whether we have forest or grass, vegetation is important. So I think what we're stuck with now is testing models that will constrain locations, and we can't really make a globally upscaled product. But Alison Hoyt did it when she was still postdoc. So this is if you just take the simple 1 pool model and upscale it, you get something like this, where on the left is the mean transit times, and on the right is Jamshid's map with the mean ages, and you can just see the difference in the scale. So one is, the mean is several thousand years on the right, and it's decades on the left. So if you want to understand the carbon cycle, I think the one on the left is better. Okay, so what we're also trying to do is convince people that we should be measuring distributions, not means, and we should be modeling distributions, not means. And so this is our transit time distribution from Harvard Forest. Sorry, that's not from Harvard Forest, it's from another one. But, and this is from Jim years ago. We talked about this many, many years ago. It was one of the impetuses for writing the proposal in the first place. But if you run like an impulse response function, you get basically a transit time distribution that you can compare directly to our transit time distribution from our 2 pool model. So we can start to understand why they might be different. The other thing we've been doing, another one of my students, Shane Stoner, just finished his PhD doing thermal oxidation of soils to actually get a distribution of the ages. And this is, on the left is he did some fractionated soil and then he added them all together to produce the blue curve on the right. Then the red curve on the right is actually a model, two-pool model prediction of the radiocarbon density distribution in the same soil for a specific year that it was sampled. So the models also could be used to predict the radiocarbon distribution, and we can start doing Bayesian fitting of the distributions, not just the means, right? So the mean, in this case, the mean radiocarbon really doesn't tell you that you have this younger and older pool. And here we can at least support the idea that two pools in the model is giving us more or less the right shape of the distribution. Okay, so although our money is finished for C14 constraint, we just put in the final report. I just want to highlight, we're going to have a radiocarbon short course with Ted Schur in Arizona, Northern Arizona University in August this year. People are interested. And we usually have a pre-AGU hackathon for ISRAD, and so this year will be in San Francisco. If you want to welcome, it's usually the Thursday, Friday, Saturday, Sunday before AGU. And I just want to finish by thanking the WMCAC Carbon Cycle Accelerator Mass Spectrometry Facility, where a lot of the data that you saw were produced, not all, and actually there's a growing amount that's being produced in other labs, but certainly all my data were produced here. And so in honor of that, and because many of you probably never have even seen the accelerator, how many of you have seen the accelerator mass spectrometry? Okay, a few of you, but not everybody. So we're gonna have the refreshments downstairs after the talk today. It's better here. So sorry I went a little long, but thanks for listening. And ideas of how to get around our model conundrum, certainly welcome. Yeah.
So this mineral hypothesis, is this that organic material is absorbed onto these minerals and biology can't access it? Is that theory? And what's the theory? Can you explain the different substrate relative effectiveness?
Yeah, so the different substrates, it's 2-to-1 clays, 1-1 clays, crystalline and non-crystalline minerals. So it's just basically the sorted surface area and strength of absorption, whether it's ionic or minerals.
It makes sense.
It makes sense, that's his thesis is about how that makes sense. What we were hoping is that it wouldn't matter if the wood is getting inspired. And what it is the DOC gets sorbed and desorbed, and when it's desorbed, the microbes get eat it. And the other thing it could be as you go deeper in the soil, one of the questions is, why does carbon get older as you go deeper in soil? And there's two answers. One is it's produced in situ and there aren't so many microbes there, so it sticks around for a long time. Or it could take that long to get there from the surface. So his other thing is he actually put a lifetime into the models he was using. And as you go deeper in the soil, the lifetime that you require to model the data gets older. So it's about a centimeter per year. So yeah, to get to 30 centimeters, it's going to take 3 decades from the surface.
Is older below that? Like they're slow, much slower moves below that.
Well, there's so little carbon at that time moving down. I mean, it's just, every time it desorbs, the microbes, take that cut, and then what's left over can move down. So we're still not completely sure, but it's very, when Jim Karashi was here, we published this paper saying we think there's a timeline that you have to explain the B horizon of the soils. If you have the time series, you can see it going up when the rest of the world is coming down. Yeah, so he has like 27 replicated sites where he can say that that's happening. It's a much stronger story now. But
This is relatively easy to test this agricultural soil, but it's actually lower. Are we saying for the depth scale?
yeah, so one of my favorite talks of the last while is that one of the treatments in New Zealand is they're flipping soils upside down to sequester carbon. It's the best way to do it. You have to flip the soil. But yeah, I mean, I left cultivated soils out of here. So there's in the ISRAD database, we have experiments, we have cultivated soils, we have everything else, but you can actually see if you put the cultivated soils in there, they tend to be aspiring over carbon because of what you said, that you're mixing, you're disturbing things, you're releasing carbon from minerals that may have been stuck on them inside aggregates, and you're also bringing older parts of the soil to the surface where they decompose quickly. Yeah. Yes.
So the fractions in Israel, are those mostly like physical separations? So I wonder if there's any like chemical separations that might get past full.
I mean, water extractable, but that's actually, they're all about the same. There's one paper, synthesis paper that kind of came out. So we have everything, we just take what's in the literature, but the most common ones are these density separations. I'd say then there's some chemical treatments of the bulk of the mineral soil that people are looking for what's stored to which strength. But yeah, they get hard to compare because that's why I like incubations. They're very simple, they're pretty robust. You don't have to do anything else, and that's why we also like the thermal methods, because you just put it in an oven and capture the CO2, and you've got the complete spectrum of ages. So the problem is when I put a leaf in, I would get a spectrum. I would get carbon released at all those temperatures too, so I had to get rid of those. This free light fraction before combusting the rest. Soils are not easy. But litter is important, and nobody's paying attention to it. It's very simple. We can do one pool model. It's totally like, so for soil scientists, it's above the soil, and for ecologists, it's below the vegetation. So everybody knows it. Sorry. That's all.
Is there any place where you have a long time series of flux towers where you could see low frequency accumulation of carbon and then subsequent release to try to model those timescales?
Yeah, no, we're assuming everything's at steady state when we do our models. But if they're not at steady state, if they're so close, it doesn't make much difference for us. I mean, we have modeled non-steady state systems. We don't have to model them steady state. We do know how the carbon change as well. So in all these sites where we use the steady state, we know it's a good assumption.
Well, my question was more, could you get a timescale that way?
So the earliest Radiocarbon towers are 1990 something, 1980 something. So they're short compared to the 100-year timescales. And I think there's lots of other things going on that you know your fields out, like climate, the oscillations of climate and things like that. We need several decades to get enough time. Okay. So